Antimicrobial resistance. It is unresponsiveness of a microorganisms to an AMA, and it is similar to the phenomenon of tolerance seen in higher organisms. When bacteria are no longer susceptible to the action of antibiotic, the bacteria are said to have developed resistance to that antibiotic. Antimicrobial resistance, or AMR, occurs when microbes evolve mechanisms that protect them from the effects of antimicrobials. Antimicrobial resistance is a subset of AMR that applies specifically to bacteria that becomes resistant to antibiotics. We have here a diagram that shows the difference between non-resistant bacteria and drug-resistant bacteria. As you can see, non-resistant bacteria multiply and upon drug treatment, the bacteria die, while the drug-resistant bacteria multiply as well, but upon drug treatment, the bacteria continues to spread. Antimicrobial resistance is mainly caused by the overuse of antimicrobials. This leads to microbes either evolving a defense against drug use to treat them, or certain strains of microbes that have natural resistance to antimicrobials becoming much more prevalent than the ones that are easily defeated with medication. While antimicrobial resistance does occur naturally over time, the use of antimicrobial agents in a variety of settings, both within the healthcare industry and outside, has led to antimicrobial resistance becoming increasingly more prevalent. There are two types of resistance: the natural and acquired. Natural resistance: the organism has never responded to the AMA, maybe due to the absence of the particular enzyme or target site affected by the drug. This is generally group specific. Examples: gram-negative organisms are not affected by penicillin G. The E. coli bacteria are naturally resistant to benzylpenicillin. They are unable to penetrate cell wall. And tuberculosis is sensitive to tetracyclines. Note that this type of resistance does not produce any clinical problems as alternative drugs are available. We have here a CDC infographic on how antibiotic resistant, a major type of antimicrobial resistance, happens and spreads. Antimicrobial resistance can evolve naturally due to continued exposures to antimicrobials. Natural selection means that organisms that are able to adapt to their environment survive and continue to produce offspring. As a result, the types of microorganisms that are able to survive over the time with continued attack by certain antimicrobial agents will naturally become more prevalent in the environment, and those without the resistance will become obsolete. Some contemporary antibiotic resistances have also evolved naturally before the use of antibiotics or human clinical use of respective antimicrobials. For instance, methicillin resistance evolved a pathogen of hedgehogs, possibly a co-evolutionary adaptation of the pathogen to hedgehogs that are infected by a dermatophyte that naturally produces antibiotics. Acquired resistance: when previously sensitive bacteria become insensitive or resistant to an antibiotic, the process is also known as acquired resistance. This is due to replacement of sensitive bacterial population by resistant population. The acquired resistance is through genetical or biochemical mechanisms, indiscriminate use of antibiotics or improper selection of antibiotic under dosage for treatment for longer shorter period than required promotes acquired resistance. This poses a clinical problem in veterinary practice. Some bacteria acquire rapid resistance, example, Staphylococci, coliforms, and tubercle bacilli. Bacteria acquire resistance by a change in their DNA. Such DNA changes may occur by mutation and gene transfer. Mutation is a stable and heritable genetic change that occurs spontaneously and randomly among microorganisms. It is not induced by the AMA. Any sensitive population with few mutant cells require higher concentration of AMA for inhibition, and these are selectively preserved and get a chance to proliferate when the sensitive cells are eliminated or lack of competition. Mutation may be single step and multi step. Single step is a single gene mutation may confer a high degree of resistance. It emerges rapidly. Example, enterococci to streptomycin and Staphylococcus aureus by rifampicin. Multi-step, where several gene modifications are involved, it develops gradually. Example, resistance to erythromycin and tetracyclines. Gene transfer. Many bacteria contain extrachromosomal genetic material called plasmids in the cytoplasm. These carry genes coding for resistance called R factors in which these R factors are transferred to other bacteria and spread resistance. Gene transfer from one organism to another can occur by conjugation, transduction, and transformation. Conjugation is a sexual contact through the formation of a bridge or sex pilus common among gram-negative bacilli or the same or another species. This may involve chromosomal or extrachromosomal DNA. The gene carrying the resistance transfer factor is also present. Conjugation frequently occurs in the colon where a large variety of gram-negative bacilli come in close contact. Even non-pathogenic organisms may transfer R factor to pathogenic organisms, which may become widespread by contamination of food or water. Transduction is the transfer of gene-carrying resistance through the agency of a bacteriophage. Example is a virus that infects bacteria. The R factor is taken up by the phage and delivered to another bacterium which it infects. Certain instances of penicillin, erythromycin, and chloramphenicol resistance have been found to be phage-mediated. Transformation is a resistant bacterium may release the resistance-carrying DNA into the medium and this may be imbibed by another sensitive organism becoming unresponsive to the drug. This mechanism is probably not clinically significant. Cross-resistance. It is the resistance seen among chemically related drugs. It is when something develops resistance to several substances that have a similar mechanism of action. For example, say a certain type of bacteria develops resistance to one antibiotic. That bacteria then also has resistance to several other antibiotics that target the same protein or use the same route to get into the bacterium. A real example of cross-resistance occurred for nalidixic acid and ciprofloxacin, which are both quinolone antibiotics. When bacteria develop resistance to ciprofloxacin, they also develop resistance to nalidixic acid. Because both drugs work by inhibiting thiopolymerase, a key enzyme in DNA replication. Due to cross-resistance, antimicrobial treatments like phage therapy can quickly lose their efficacy against bacteria. We have here the illustration of antimicrobial resistance mechanisms as microbes adapt to stand resilient against the effects of the drugs and other agents meant to destroy them.